Hello, welcome to the Tissue Mechanics and Mechanobiology Laboratory at WPI. I'm Professor Chris Billiar, and I'm a biomedical engineer. Our group works to understand how physical forces affect cells in the body, known as mechanobiology, a relatively new area of research in biomedical engineering. It's actually an old concept, like building muscle when we exercise and losing it when we act like couch potatoes. More broadly, all our biological tissues, like our skin and our bones, are subjected to forces from gravity, muscle contraction, blood flow in arteries, and air in our lungs, and even the food in our stomachs. These signals and forces signal the cells to grow and even to die, depending upon the need.
Hello everyone, this is Ying. Heart valves regulate our blood flow and prevent backflow. The valves open and close 2.5 billion times within our lifetime. As shown in the cartoon, healthy valves can open and close fully. However, in diseased valve, due to calcification and thickening, they cannot operate properly. Unfortunately, heart valve disease is a major cardiovascular disorder, resulting in 300,000 replacement surgeries worldwide each year. Tissue-engineered heart valves are now in development based on patients' own cells. Therefore, they can reduce the immune responses, repair themselves if necessary, and grow with patients.
The ultimate goal of tissue-engineered heart valves is to have them fully integrate with the body. When you're an embryo, your valves develop when cells in the area secrete and reorganize structural proteins in a process known as remodeling. Researchers want to mimic that process in tissue-engineered heart valves in order to obtain a healthy environment. And this includes getting cells from the blood to stick on and migrate into the engineered tissue to initiate that remodeling process. Our lab uses miniature models of tissue-engineered heart valves that we call Draeger tissues, and they can allow us to evaluate mechanobiological responses more clearly. For example, we can look at how the behavior of cells and their remodeling changes with hemodynamic forces, similar to how you would see in the valve environment. We can place the microtissues under mechanical stretch to mimic the hemodynamic forces in the valve.
We made some microtissues on top of soft silicone rubber posts. The cells in the tissues apply contractile forces to bend the posts. After culturing tissues for 4 days, we fixed and stained tissue sections. We can see the dark spots, which are nuclei of cells. Also, we can see the blue color, which are gags in the tissues. The pink color shows collagen secreted by the cells.
Hi, my name's Sophia Noel. Our lab has developed a device that quickly and easily calculates the exact stiffness of these PDMS posts. If we know the exact stiffness of the PDMS, we can calculate the forces on the tissue while it's growing and how that impacts the contractile forces of the microtissues. Knowing this can then help us better determine which mechanical conditions result in ideal cardiac tissue properties. This knowledge can be applied to create protocols to grow better tissue-engineered heart valves.
Hi, this is Mahva Shebeli and I talk about the model I am using in my project, which is investigating the role of apoptosis in calcification of the aortic valve. Why calcific aortic valve disease is important? It is a significant source of mortality in the US. 25% of people aged more than 65 years old are dealing with it. And if it is left untreated for more than 5 years, mostly leads to heart failure or death. To study the behavior of the cells, the simplest way is in a controlled environment, such as in the laboratory, where we can keep all the conditions the same and change one of them as our variable and study its effect on the cells. Also, the behavior of the cells as single cells differs from their behavior as a group of the cells. That is why it is important to study them as a group that are in contact with each other and influence the collective behavior of the group. With the help of this model, we hope to investigate the causational relationship between the cell behavior.
The stiffness of a cell can tell us many things. A stiffer cell is usually more contractile, like a trampoline stretched by many tightened springs. It has a larger number of stress fibers than the softer cells. The stiffness of a cell is a hallmark of many physiological changes of the cell. For valvular interstitial cells, Low cell stiffness is related to excess cell death and the calcification of aortic valves. We poke cells with a microbeads as small as 10% of the thickness of a thin hair to sense their stiffness. Cell stiffness is also related to cell movement. Cancer cells are usually softer than the normal cells, which makes them easy to move around the body to form new tumors elsewhere. Cancer cells are also of varied stiffness. The softer cancer cells are more invasive than the stiffer cancer cells. This relationship may help doctors to predict the potential of a tumor to spread in the body.
Hi, I'm Habibe. In our lab, we use several tools to measure mechanical forces and compute the stresses within the tissues and cells. The effect of these stresses on biological phenomena will then be studied. Here, we are preparing the instrument testing machine to do anisotropic indentation on tissue. We then use the data to extract tissue mechanical properties using finite element modeling and machine learning.
Our lab includes and appreciates people of all backgrounds, races, ethnicities, and gender identities, and includes postdoctoral fellows, graduate students, undergraduates, and even high school students. We hope to see you in person soon.